Next up, we're going to learn about how the hormone ADH, antidiuretic hormone, helps to regulate water reabsorption in the collecting duct. So remember, we learned that water can get reabsorbed throughout the nephron, different locations. In fact, most of it is reabsorbed in the proximal tubule. However, the one place where it can be regulated by a hormone is in the collecting duct. So let's take a look at that first. So imagine this is the collecting duct, our nephron is over there, and we have the vasa recta, the capillary surrounding the collecting duct, and so we can do reabsorption. So let's pretend that we're in a situation where we are dehydrated. So would we want to do more water reabsorption or less water reabsorption in that situation? So remember that reabsorption is the movement of a molecule from the nephron into the blood, like back into the blood. So if we're dehydrated, we want to hold on to that water. We don't want to lose it in the urine. So we actually want to increase water reabsorption, right? We want to move that water back into the blood to make sure it doesn't end up in the urine, which is what's going to be made at the end of the collecting duct here. Okay, so how can we help regulate water reabsorption? How can we increase water reabsorption? So remember that water is a polar molecule and it needs to go through a channel in order to get across the plasma membrane. And so what we can do is we can regulate the aquaporins, the water channels in these membranes here, and that will regulate water reabsorption. And so that's in fact exactly what this hormone ADH does. So ADH is released when we're in these dehydrated situations. And ADH, you may recall, is actually released from the posterior pituitary. So I have that drawn over here just to remind you about how that process works. We went over this such a long time ago. So this is the hypothalamus. This is the pituitary gland, part of the brain. And remember that for the posterior pituitary, it's just a one-step process. There are no endocrine cells. It's actually a neuron that has its cell body in the hypothalamus. Its axon goes down into the posterior pituitary. It releases ADH, that's a peptide hormone, so it's lipophobic, and that is released into the blood. It's also a neurohormone, right, because it's released by a neuron. Remember when you first saw this diagram and you maybe didn't even know what a neuron is, but now you know, right? So an action potential in the hypothalamus neuron causes the release of ADH via exocytosis of a vesicle, so think about how much that you've learned. Okay, so now this ADH is in the blood, and it's going to travel back to the collecting duct and increase the number of aquaporins there. So let's look at how that works. So we're just going to basically zoom in on this. So let's look at like one cell or a couple of cells that are in the wall here of the collecting duct. Okay, so I'll just draw a couple of cells here. These are epithelial cells, right? So I'm drawing them with little microvilli on them. Something like that. Okay, so these are the epithelial cells that are found in the collecting duct. This would be the lumen, so the filtrate would be over here. So this is the lumen of the collecting duct, so in other words, like right in here, right? And so I'm looking at these cells here. And then on the other side would be the capillary, right? So the vasa recta is over here. Okay, so this is our vasa recta. These are our collecting duct epithelial cells. We'll write that in here. So collecting duct epithelial cell. All right, cool. So remember ADH was just released into the blood. Does it need a carrier protein? No, because it's lipophobic, right? So it's a protein, it's a peptide. So ADH is just traveling here in the blood, comes along. Capillaries are leaky, so ADH is just going to leak out at some point, and it's going to bind its receptor that is in the membrane of these collecting ducts. Okay, so let's put in a receptor here for it. Okay, so ADH is just going to travel out here and bind to its receptor. So this is the ADH receptor. Note I drew it in the membrane because we know that this is a lipophobic hormone, and so it needs to have a membrane receptor. Now this receptor could be either enzyme linked or it could be a GPCR. So remember, those are the types that are used for hormones. It turns out that it's actually a GPCR, and it activates our old friend, the cyclic AMP pathway, right? So this G protein is going to activate our enzyme, adenylate cyclase. I'm just going to write AC for adenylate cyclase. That converts cyclic AMP into, ATP into cyclic AMP. Said that the wrong way. Okay, so we're going to make our second messenger cyclic AMP, and then cyclic AMP is going to activate pKa. This should all sound very familiar. So this is our kinase enzyme. And now the next part is pretty interesting. So there are stored inside of these epithelial cells are vesicles, and these vesicles have aquaporins in the membrane. This is actually a very common strategy for cells. So if a cell needs to put more channel or receptor or transporter or something into the membrane, often it will store it in the membrane of vesicles. So let me label that. So these are aquaporins. And right now they're being stored just in the membrane of these vesicles. All right, so when pKa gets activated, it's going to phosphorylate proteins that are in the membrane of these vesicles that cause the vesicles to do exocytosis. We don't need to worry about exactly how that works, but it's going to cause this vesicle to fuse to the membrane, right? And usually we've talked about exocytosis in the context of releasing a neurotransmitter or releasing a hormone. In fact, that's how ADH was released. But in this case, we're not releasing stuff that's inside of the vesicle. We're just trying to get these aquaporins into the membrane. And so when we do exocytosis and the vesicle fuses to the membrane, now our aquaporins are in the membrane, right? So we've just added a bunch of aquaporins to the membrane via exocytosis of these storage vesicles. All right, so now we have aquaporins here. And so what does that mean? That means that water that's in the lumen here can now enter inside of our cell. Okay, so now it's inside the cell, but not quite in the blood yet. So we need to have one more thing, which is that on this side of the membrane, there's actually already aquaporins. So it's a different type of aquaporin, and it's always here. Okay, so water goes through our first set of aquaporins, and then it's going to go through our second set as well. Right, and so we just did reabsorption of water. All right, so maybe we should talk through that one more time. Okay, so when you're dehydrated and blood osmolarity is high, like concentrated, that signals ADH to be released. We'll go over how that happens in a second. ADH then travels through the blood here. It makes its way to the vasa recta in the kidney. ADH comes out, it binds to its receptor, which is a GPCR. So you see a G protein there. That activates the second messenger system, cyclic AMP. So cyclic AMP activates pKa. pKa phosphorylates a protein in these vesicles that causes exocytosis. So this arrow here is exocytosis. And now this aquaporin is being put into the membrane, right? So we're increasing the number of aquaporins in the membrane on this side, the side that's facing the lumen, the filtrate. And that allows water to enter the cell. Water can then go through the other membrane because the aquaporins on the other side are always there. Okay, and that's fine. It doesn't matter if they're always there or not, because what we need to regulate is water going from the lumen into the cell. Right? Once it's there, then for sure it's going to go into the vasa recta, and that is water reabsorption. So we're holding on to the water that we have so we don't lose it in the urine.